Assalamu alaikum. I have a question for you. Have you ever been to a factory or you observe the internal environment of a factory? These are the pictures which actually explain you the internal environment of the factory. Here you find different people working different performing different jobs. They are specialized people. They are the main component of the factory. What happens if these workers are not there or they are absent? What do you think? The factory will produce the same amount of products if they are absent? Yes, you are right. It will not be possible. The labor are very important for the factory. In the same way, your cells are actually a chemical factory. In the cytoplasm of the cells, lots of chemical reaction takes place. And these chemical reactions are under the control of a special labor. And these labor actually speed up the rate of reaction or speed up the chemical reactions. Today, we will discuss about these levers. The name of the lever here, enzymes. I am Sir Faizan and today I will discuss some topic related to the enzymes. These are the learning outcomes of this topic. First of all, we will discuss about catalyst and enzyme. What is enzyme? What is catalyst? The second objective, characteristics of enzyme. What properties of the enzymes? Third, we will discuss or explain enzyme action in terms of a special hypothesis which is known as lock and key hypothesis. The fourth learning outcome is describe the effect of temperature and pH on enzyme activity. Let's start. First of all, catalyst. In many chemical reactions, a catalyst is added to the reaction mixture. A catalyst is basically a substance or a chemical that speeds up the reaction but is not used up in the reaction. It is a speciality of the catalyst. A catalyst is neither a reactant nor a product. You can see the equation here. H2O2 is one of the compound which is known as hydrogen peroxide. This compound produces two chemicals, H2O water and O2. This H2O2 is known as reactant, and after the arrowhead you will find the two products. This H2O2 produce H2 and O2. On the arrow you will find MnO2. This MnO2 is actually a catalyst. As I have told you, it is a substance that speeds up the reaction but is not used up in the reaction. It means that this MnO2 remain in the solution without any change. But the compound, the reactant H2O2 will convert into H2O and O2. So a catalyst is neither a reactant nor a product. So its formula is written above the arrow in a chemical equation. Normally in chemistry laboratories or industries, you will find many catalysts and they are directly involved in the speeding of the chemical reaction like liver in the factory, which actually accelerate the production of the different chemicals in the industry. What is enzyme? Enzymes are actually proteins that speed up the chemical reaction in our cells. Or actually, enzymes are biocatalysts. As I have told you, catalysts are the substance which speed up the reaction. So enzymes also speed up the reaction, but these catalysts are present in your cells, in animal cells, in plant cells, or even bacteria cells. So enzymes are also known as biocatalysts. Bio means living. So they are present in a living bodies. What about the nature of the enzyme? They are protein in nature. The molecules, the organic molecule protein, they are made up of protein molecules. And the speciality of this protein, it is speed up the chemical reaction. In this slide, I have an example for you. Your liver cells is very important. Liver is one of the largest organ in your body. Largest organ in the sense largest gland in your body. This liver produces the enzyme known as catalase. This is catalase, and this catalase is involved in speeding up the breakdown of the hydrogen peroxide. Hydrogen peroxide is broken down or convert into water and oxygen with the help of this enzyme catalase. If this catalase is absent or will not produced by the liver, so this hydrogen peroxide is actually one of the poisonous chemical. So it will destroy or disturb the cells of the liver. So the timely breaking of the hydrogen peroxide is very important, and this timely breaking depend upon the presence of the catalase, the enzyme. So catalase quickly break down this compound and convert into H2O and oxygen. So the chemical that the enzyme act on are called substrates. So here the catalase act on the hydrogen peroxide. So this hydrogen peroxide known as a substrate. The substance produced are called product. So in this equation, the two products are formed, water and oxygen. So they are known as products. This is what in this first slide. Now we are moving towards the characteristics slide. Characteristics of the enzymes. The first characteristic of enzyme is enzymes are protein. What is actually protein? Their folded chain of amino acid can be represented by simple shapes. This individual circle represents an amino acid. So a long chain of amino acid is actually known as protein. So this is a protein molecule. And the shape of the protein molecule, we represent enzyme, we draw enzyme in this shape. So the protein structure is actually represented by the help of this shape. Now I am going to show an animation where you find a reaction which is completed with the help of an enzyme, and again the same reaction without an enzyme, so that you can understand what the enzyme do with the reaction. The first animation for you, where we see the difference in a reaction with or without enzyme present. To understand how enzyme help in chemical reaction to occur more quickly, we will view a simple synthesis reaction. First of all, we will select a reaction which is happening without an enzyme. These two simple sugar molecule can combine to form a large double sugar molecule if they come together at just the right angle. You can see here the structure of this sugar molecule, and this is again a sugar molecule. This is a free end of a sugar molecule which can join the free end of another sugar molecule so that a big molecule can form. Look at the collision of these molecules. The molecules are colliding with each other, but they are not in a correct orientation so that they can make a bond. It's still two simple sugar molecule they didn't make their bond with each other. Now see what happened, the same reaction with the presence of the enzyme. This big yellow structure is actually an enzyme, and you will find some depression or pockets in the structure of enzyme. These pockets or depressions is known as active site. Again, you will find the two sugar molecules, simple sugar molecules. Now, the enzyme shown here is a specific for this reaction. Only these simple sugar molecules will fit it, and they can attract it to the enzyme. You can see the shape of this active site. It is almost similar to the shape of the simple sugar molecules. Now, when a simple sugar molecule fit against the enzyme, they are oriented properly to react and make a larger molecule. So this orientation with the enzyme actually help the sugar molecules to make a bond. So now you can see that the free end actually connect with each other or link with each other and form a molecule which is bigger than simple molecules. In this way, enzyme perform its reaction, and this is known as the bond. So enzyme actually involve in the formation of two small molecules, the formation of a big molecule by the help of the combination of two small molecules. This is known as anabolic reactions and which are normally takes place inside your cytoplasms. The new double sugar molecules leave the enzyme is unchanged and can help two more molecules to react. So in this animation, you understand that the molecules which are going to make bond require some proper orientation. And this orientation, the gaining of the orientation require time. Enzymes actually minimize the time by giving the correct orientation to the molecules. In this way, enzyme is speeding up the reaction. And the second very important thing in this slide, you observe that there is no change in the structure of the enzyme. It means enzyme remain the same. It is unchanged. It do not alter. So we have understood that enzymes are actually involved in chemical reactions, but they do not alter, but they speed up the rate of reaction. Now the second characteristic of enzymes. Enzymes are specific for specific substrate. This specificity depend upon the shape of the enzyme. Enzyme act on a substrate by attaching to them. The part of the enzyme to which one or more substrate molecule can attach is called the active site. You can see this diagram. Here you find the active site. This active site is the site where substrate can easily combine. Now you will find the three enzymes at the base of this slide: enzyme type 1, enzyme type 2, and enzyme type 3. Enzyme type 1 has a specific substrate of that particular enzyme because the active site is different. So the substrate which is similar to that active site shape, which is similar to the shape of this active site, can easily bind with this enzyme. So this enzyme is, utilize the substrate or act on the substrate. The enzyme 2 is the active site is somewhat triangular in shape. So any substrate which is triangular in shape can easily fit in this active site. So enzyme 2 is, is specific for a triangular type of or triangular or substrate which is triangular in shape. Enzyme 3 is again has its active site. It is not triangular, it is somewhat hexagonal. So any substrate which has hexagonal shape can easily combine with enzyme type 3. So every enzyme has its has its active site which is of different shape. So on the behalf of that shape, the substrate which is similar to that shape, active site shape, can easily fit in. So in this way, a specificity is confirmed by the help of the active sites shape. I'm going to uh, show you another animation by which you can understand the active site and the attachment substrate with the active site. Okay, now this is a present animation where you find the nature of the enzyme and the nature substrate. In this screen, you will find two type of enzymes: green enzyme or yellow enzyme. The important thing is that these two enzymes are of different active sites. Yellow one again has two active sites but of different shape as compared to the green one. Again you will find a substrate which is a double sugar already bonded. So enzymes are specific, meaning each enzyme can only be used for one particular reaction. Now tell me, this double sugar molecule combine with which type of enzyme? Enzyme yellow or enzyme green? Okay, let's see. Oh, this this substrate will not be able to combine with the enzyme which is green because the shape does not exactly fit with the enzyme. So double sugar molecule does not fit into the green enzyme. Therefore, the enzyme cannot help the digestion. A perfect fit. This double sugar molecule can fit into the yellow enzyme so it can help the sugar molecule to be digested. It is actually the active site which is similar to the substrate molecule. That's why it's perfectly fit in this one. Now, the double sugar is converted into simple sugar. And the digestion is basically the conversion of a large molecule into the simple molecule. So it is one of the example of digestive enzyme. Here we can see a reaction that might use the green enzyme. A different sample sugar molecule have a different shape and would not fit the yellow enzyme. You know, carbohydrate are of different type. Glucose, fructose, galactose, these all are sugar molecules. But glucose is different from fructose and fructose is different from galactose. So here in the slide, you will find again the two molecules of the simple sugar but of different shape. It's mean they are sugar but different formula. Yes. Now in this example, a green enzyme will work and green enzyme can attach these two simple sugar and make a bond. So green enzyme is actually acting as a synthesis enzyme. It can join two small molecule and form a large molecule. While yellow enzyme in the previous slide, as you have observed, it can break down, it can involve, it is involved in breaking down of a big molecules and convert the big molecule into a simple molecule. So the yellow one is acting as a digestive enzyme, or the green one is acting as a uh, formation enzyme, which involved in the formation of big molecules. So in this way, the green enzyme helped to form this double sugar molecule. The yellow enzyme doesn't have the specific shape needed to have this reaction. Thank you. Now, Alhamdulillah, we have observed that the specific enzymes react on or combine with a specific substrate. It's because of the active site of the enzyme. So, there is a theory or a hypothesis which is known as lock and key hypothesis. The lock and key mechanism is basically explain you the interaction of a specific substrate with a specific enzyme. Enzymes are specific, only molecule with the correct shape can fit into the enzyme, just like only one key can open a lock. Only one type of enzyme can speed up a specific reaction. This is called the lock and key model. It's very simple. You can see the diagram here. The substrate collide with the active site of an enzyme and become attached. So after the attachment, they form an enzyme catalyzes, that is breakdown of the substrate. Finally, product released from the active site and enzyme again free and ready to attach to combine with another molecule. There is an effect of temperature and the pH on the enzyme. As you know very well, the labor of a factory also has its own demands and needs. They require some motivation or some kind of reward so that they can work very effectively. In the same way, your cell's labor enzymes has demand something, that is temperature and pH. Now characteristics enzymes, and that is what the temperature effect of temperature on the activity of the enzyme. Here I'm using the word heat sensitive and act on a particular optimum temperature. It's mean that enzyme need a specific temperature on which it show its maximum activity. The temperature at which enzyme show its maximum activity is known as optimum temperature. For human, it is very close to 37 degrees centigrade. It is the normal body temperature. So at your normal body temperature, your enzyme work best and show its full activity. This is a graph where you can easily understand the effect of temperature on the rate of reaction. On x-axis, you will find the temperature in Celsius. On y-axis, you will find the rate of reaction. Initially, at 0 degree centigrade, there is no activity of the enzyme. But soon, when you change the temperature, when you increase the temperature from 0 to 10, you will find the change in the activity of the enzyme. After 10 degree to 20 degree, again a 10 degrees difference, you will find the again the highest, the change in the rate of reaction. At some point, you will find the maximum peak of the graph. This is what the optimum temperature, where you find the maximum activity of the graph. This is according to this graph, it's almost 37 degrees centigrade. What happened when you further increase the temperature? The enzyme denature at a high temperature. It's mean the activity will fall. Actually, when you increase the temperature, the protein, the amino acid which form the enzyme, they actually break apart, or the bond between the amino acid will basically break. So the active site is disturbed. If the active site is disturbed, substrate will not be able to combine with the enzyme. That is the reason we use the word enzyme denature, means enzyme loses its shape or active site loses its shape. So at almost 60 degrees centigrade, enzyme denatured. So enzyme will not be able to perform its function. It is useless. Again, the second important property, or which basically enzyme are sensitive to that property, is basically pH. What is pH? P for power, H for hydrogen. Power of hydrogen ion in a solution is known as pH. This pH is a scale has a value from 0 to 14. From 0 to 6.9, it is acidic pH, 7 is neutral, and from 7.1 to 14 is actually alkaline pH. Change in the pH also alter the enzyme shape. That means enzyme active site shape. Different enzymes work best at different pH. Normally, the optimum pH for an enzyme depend upon where it normally works. For example, intestinal enzyme have an optimum pH of about 7.5. Enzyme in the stomach have an optimum pH of about 2. Can you tell us what is the optimum pH of this enzyme with the help of this graph? The maximum peak is actually indicate the optimum pH. So here you find 8 or something, or somewhat below 8, 7.8 or 7.9 is basically what the optimum pH where enzyme show its maximum activity. Again, increasing the enzyme activity on the y-axis, and on x-axis you will find the pH changes. Again, you find this on both sides of the optimum pH, you will find the activity will decrease. So enzyme need an optimum pH, a specific pH on which it show its maximum activity, and this is known as optimum pH.